All living things are composed of eukaryotic cells. Now, there are a lot of factors that need to be considered when we think of the word cell. First, we'll take a look at what these structures look like. Then, we'll dive deep into the detailed workings of the cell and which components perform which functions. Then, we'll uncover the hidden differences between a plant and animal cell and finish off with a look into one of the most important parts of the cells, the membrane. This is an animal cell. It's the cell building block of, you guessed it, animals. It's known as a eukaryotic cell due to its nucleus in the center, but we'll talk about that later. Now, before we get too far, know that some cells develop cilia or flagella on their extremities. These can look like tails or like a bunch of tiny little arms. Made out of microtubules, these components help move the cells or liquid around the cells. Now let's take a look at the cell membrane, which encloses the entire cell, which is a lot more complex than this shape, but we'll get into that. The cell membrane protects the integrity of the cell's interior by allowing certain substances into the cell and not allowing others, thanks to its selective permeability. Within this enclosure, a solution of water and nutrients will be found called the cytoplasm. Within the cytoplasm is the cytoskeleton, and this is basically various protein strands that aid in enforcing the cell. The cytoplasm gives a sort of infrastructure to allow all the cell components to work their magic. However, there is another type of plasm, but we'll get into that in just a second. Moving on, we'll find the endoplasmic reticulum, which is a network of membranes that carries things around the cell. There's two types of endoplasmic reticulums, ER for short, the rough and the smooth. The rough ER specializes in manufacturing lipids, or fats, and proteins. This network is called rough because it is dotted with ribosomes, which are spat out from the nuclear envelope, and they make up the proteins that function in the cytosol. The smooth ER, on the other hand, is more known for synthesizing lipids and lipid-containing molecules, such as the phospholipids that make up the membrane. Amazingly, it can also help detoxify drugs and alcohol in the liver. Amino acid chains are formed by ribosomes, are sent off in the ER, and then are sent off to the Golgi apparatus. The Golgi modifies lipids and those protein chains received from the ER and packages them into vesicles, which also have phospholipid walls, and then ship them out to other parts of the cell or out of the cell. The Golgi bodies make up these apparatuses, and they kind of look like stacks of flattened green pancakes. The Golgi bodies also help finalize the lysosomes, which are small sac-like structures that contain a strong digestive enzyme that, when released, can break down worn-out organelle in food. Now, the nucleus is known as the brain of the cell, and it contains majority of the cell's genetic DNA in the form of DNA molecules and protein. The nucleus is able to store and replicate the genetic information of the cell. The nucleolus is found in the nucleus and contains RNA, protein, and chromatin, and spits out ribosomes. And separating both of these components from the rest of the cell is a double membrane nuclear envelope, so you could say the nucleus is fairly protected. And here's where the second plasm comes back in, and rather than floating around in the cytoplasm, the nucleus finds itself in the solution of nucleoplasm. And finally, the power plant of the cell, the mitochondria. Round tube-like organelles with their inner membranes highly folded. Energy is derived from carbs, fats, and other fuels and is converted into ATP thanks to the mitochondria. But wait, I almost forgot. This was the animal cell, and although very similar, the plant cell has certain characteristics that it does not share with its close counterpart. One differentiating factor is the cell wall, which gives the plant cell that more boxy shape compared to the round one of the animal cell. The wall is mainly made out of cellulose and lignin, which are both two pretty tough compounds. Next, plants have plastids, which are organelles that make and store the compounds that they need. The most important of these are the chloroplasts, which convert light energy from the sun into sugar and oxygen. Also, there's a large central vacuole. Plant cells push water into vacuoles, which provides pressure, reinforcing the already strong cell wall. While some animal cells do also have vacuoles, they aren't as large as the central vacuole and they aren't tasked with keeping the shape of the cell. So now that we know the components that are in the animal and plant cells, let's take a deeper look into the specific differences in structures of both cells. Firstly, the size. Animal cells, generally 10 to 30 micrometers in length, are smaller than the 10 to 100 micrometer long plant cells. Their shape differs as well, as the animal's is more rounded or irregular, and the plant cell is rectangular or cube-shaped. They also store energy differently. Plants store energy as starch, whereas animal cells store it in the form of complex carbohydrate glycogen. Finally, both cells grow differently. Animal cells grow by increasing in numbers, where plant cells grow by absorbing water into their central vacuole. Now that we know the plant cell, the animal cell, and every difference in between, let's dive into the cell membrane. Primarily made up of phospholipids, cholesterol, and protein, membrane lipids are held together by weak intermolecular bonds rather than strong covalent bonds. This allows the molecules in a membrane to move about freely. Phospholipids in a bilayer exchange places millions of times within a single second. If a puncture or tear occurs in a membrane, molecules will quickly rearrange themselves to seal the rupture. How does it do all this? Well, let's talk about the composition. First, the phospholipid. There's the polar head, the phosphate molecule, and the nonpolar tails, the fatty acids. They assemble in their bilayers so that the tails face the inside, whereas the heads face the aqueous solution on either side. This is called self-assembly. Next, cholesterol. It's a type of steroid that's helpful in regulating molecules entering and exiting the cell. And thirdly, protein. The cell is made up of two different types of proteins. Integral proteins are nestled into the bilayer and stick out on either side. They are helpful with transporting larger molecules. Then there are the peripheral proteins, which don't extend across the membrane. They can be attached to the end of integral proteins, or not, and help with transportation and communication. Well, what makes it fluid? Well, the movement of the mosaic of the molecules makes it impossible to create a completely impenetrable barrier. Temperature has an effect on the fluidity, as when it's hot, the phospholipids will be found far apart and they'll be too fluid and won't hold their shape. But when it's cold, they'll be really close together but will be too rigid and might break. Cholesterol also has an effect, as they hold the phospholipids together so they don't separate too far, leaving large gaps. Finally, being a saturated or an unsaturated fatty acid has an impact. The tails of phospholipids are made out of fatty acids. Saturated fatty acids are made out of a carbon chain with a single bond in between them, making the tails straight. However, unsaturated fatty acids have double bonds in between them, which creates kinks in the chain, making them harder to pack in tightly and seamlessly. And finally, let's take a look at how substances get transported across cell membranes. We'll begin with methods of passive transport, meaning that molecules or ions are moving across membranes without needing energy because they're flowing with the concentration gradient. So, in the process of passive transport by diffusion, a substance tends to move from an area of high concentration to low concentration. Picture you're spraying Febreze into a large room. The area where you're spraying will have a higher concentration of Febreze molecules, and the rest of the room will have a lower concentration. Now, it's far more likely that the Febreze molecules will move from the more concentrated area to the lesser, rather than the other way around. Like a cell, the movement of molecules will continue as such until the concentration becomes equal. Next is passive transport by osmosis. Now, it's very similar to diffusion, but the concentration shift happens with water. Water molecules move because the membrane is impermeable to a solute, and the solute concentration may differ on either side of the membrane. Water regulation is crucial because if too much water leaves the cell, it shrinks, but if too much water enters the cell, it swells. Thirdly, let's take a look at passive transport by facilitated diffusion. Now, this is still a method of passive transport, but instead it's for molecules that have a harder time passing through the membrane. A membrane protein, such as a channel or a carrier, is used for this. A channel protein forms a channel along the length of the membrane, allowing ions or polar molecules to travel along their concentration gradient across this membrane. The carrier protein is similar, but it actually binds to and transports one or more particles of a substance to the other side of the membrane. Moving on from passive transport, we'll find primary and secondary active transport. For primary, we can notice that the use of ATP is used directly in order to move molecules. A good example of this is the sodium-potassium pump. Sodium ions are brought out of the cell and potassium ions are brought into the cell. Both happen against their concentration gradient, so ATP is required. As for secondary, it uses an electrochemical gradient as its source of energy. For example, the hydrogen sucrose pump. Hydrogen ions are pumped out of the cell by the help of ATP. this creates an electrochemical gradient, and the hydrogen ions eventually are able to provide the energy necessary to transport sucrose across the concentration gradient. And now for the three types of endocytosis, which is defined as the process by which the cell membrane engulfs extracellular material to bring it inside the cell. First is phagocytosis. This is simply endocytosis, but with solid particles. Second is pinocytosis. This is now endocytosis, but with, you guessed it, liquid particles. And lastly, in the endocytosis family, is receptor-mediated endocytosis, which involves the use of receptor proteins that bind with certain molecules outside the cell. The area where all these receptor proteins are found is called the coated pit because of the layer of protein. During receptor-mediated endocytosis, those receptor proteins bind with molecules and the pit folds inwards to form a vesicle. Whatever remains in this vesicle can be used or not by the cell. Now, before we exit, let's learn about exocytosis. It is another transport method. However, exocytosis fuses a vacuole with the cell membrane and releases the contents outside of the cell.